All right. Hello, anatomy and physiology students. So this week we're diving into chapter 10, where we will be learning about how muscle functions at the tissue level. So in lab, we're learning about the names of specific skeletal muscles, and their attachment sites and what kinds of actions they can perform on the skeleton. But this chapter is diving deeper into the actual anatomy and physiology of those muscles. So how they work at that tissue and cellular level, how we can actually produce the contractions that end up moving the skeleton, and all of the other functions that muscles provide for the body beyond just movement. So in this first video, we're going to start off with the introduction to just characteristics of muscle, their functions, and then some basic skeletal muscle structure. Skeletal muscles are going to be our main focus through this chapter. We'll apply some of what we're learning about skeletal muscles to understand how those same things show up in cardiac muscle and smooth muscle. But for most of this, we are focusing on skeletal muscle function and how that works, right? The actual muscles in your body that you have some cognitive control of and awareness of. So all three of our muscle types have these same characteristics. They are all contractile, meaning that they can shorten. They are excitable, meaning that they react to changes in membrane potential. They can conduct electrical signals, very similar to the action potentials that our nervous tissue uses to send messages down your neurons and through your nerves to stimulate the body and sense different things. Muscles are also able to send an electrical signal along their membrane to cause other reactions within themselves. That's how we stimulate contraction. They are extensible, meaning that they can lengthen to up to 3 times the length of their contracted length. So if a muscle is fully contracted, all of the protein fibers are fully overlapping. We can extend the length of that muscle to up to 3 times that length. We can double that and even triple it. And then lastly, they have elasticity, meaning that they tend to snap back to a resting length. And that resting length may differ depending on how you have trained that muscle, but it essentially sets the equilibrium, sets the homeostasis for where that muscle wants to be. And so even after lengthening it, it will snap back into that resting position. And so those 4 characteristics apply to all 3 muscle types. But one of the major ways that skeletal muscle, cardiac, and smooth muscle differ is on whether or not you have some cognitive control and awareness of them. So skeletal muscles are all the muscles of our body that are voluntary, that you can think about and then apply some sort of control to. So a lot of the obvious ones are the large skeletal muscles that move our skeleton, like the biceps and triceps. You can think about that movement and cause it to happen. Right? I can think about flexing my elbow and my biceps will do that. But this also applies to some of our automatic skeletal muscles that you don't necessarily need to think about in order to stimulate, but you can control with your cognitive thought. And so these are muscles like the diaphragm, which helps control breathing, muscles of the eyelids that help you blink, the muscles of the throat that help you swallow. Right? Your body will naturally keep a pattern and rhythm with those things. You are breathing, blinking, swallowing without needing to be aware of and think about that. But you can also stimulate some sort of control, right? I can take a big breath in and hold that, forcefully exhale, whatever. I can force myself to blink and keep my eyes closed. I can forcibly swallow, right? You can apply some control even though it's not needed to keep those things running. So those are automatic skeletal muscles. But most of your other skeletal muscles will only function when you tell them, when your brain is actively telling them to do something, unless there is a reflex being stimulated, which is a whole other thing. So cardiac and smooth muscles, on the other hand, are completely involuntary. You do not have any cognitive control over their function. You cannot tell your heart to beat faster. You cannot tell the smooth muscles of your stomach to churn the food that you are digesting and push it into the small intestine. right? The muscles that are controlling those types of functions are completely involuntary and out of your control. Those are viscerally controlled. And so now looking at our general functions of muscles, skeletal muscles have a wide range of function. Again, we tend to focus on and think about the movement of the skeleton, which is what the name comes from, the fact that most of these muscles are attached to the skeleton. And if that muscle crosses a joint of any kind, then when it shortens, that is going to cause a movement at that joint, right? And that allows us to manipulate our bodies and move around our environment. But they have lots of other functions that you may not necessarily think about so often when thinking about what muscles are doing for your body. They are what maintain your posture. That is maybe somewhat direct. Muscles need to be contracted or in some contracted state in order to hold your body position. Right? If all of your muscles were completely relaxed, you would be a blob on the floor. Right? So muscles need to have some sort of tension to hold your body in a seated position, in a standing position, in any kind of thing that isn't fully relaxed. Right? So they're constantly maintaining posture. They're providing support and protection to your entire organism. They play a huge role in your blood sugar regulation, as your muscles are one of the main tissues in your body that are utilizing glucose, your carbohydrates, and thus are constantly pulling glucose out of the blood, and sending or playing a role in the signaling mechanisms that regulate how much glucose is available in the blood. Playing a role in whether glucose is being stored in the liver or that glycogen is being broken down so that we can release more glucose to the blood, those things. Skeletal muscles are playing a role in some of our regulation of the passage of materials from one location to another. So any connected organs are going to have a sphincter muscle in between them, which the major component of that sphincter muscle is smooth muscle. It's viscerally controlled. It's out of our conscious control. But there is a external ring of skeletal muscle around them as well to allow for some sort of conscious stimulation in some cases. And then lastly, our muscles are playing a huge role in our homeostasis through regulating body heat. And if you think about when you get extremely cold, your body starts to shiver. That is just stimulating your muscles, generating some kinetic energy, which is being released as heat, and thus helping to warm up your body and keep it from getting too cold. So our functions for cardiac and smooth muscles are a bit of a shorter list. Your cardiac muscle really just has one job. Its job is to pump blood, to keep your circulatory system going, right? But your smooth muscles are those that are regulating all of the movements of things in your internal organs and passageways. So they are lining all of your cavities, all open lumens, right? Like think about the inside of your rib cage and the inside of your abdomen, where you have a bunch of organs kind of squished into this space, but there's also space around it. So there is smooth muscle lining that space. They are changing diameter of your blood vessels and changing diameter of your airway vessels. They elevate your hair follicles. They move contents of lumen through a tract, which is kind of the function I was focusing on earlier. Right? Moving food through your digestive system, things through your urinary system, your reproductive system, all of these passageways where something in the body needs to move from one space to another. Smooth muscle is the major player in controlling that movement, right? You don't have to think about it. You aren't really aware of when and how those things are moving. That is all being controlled by mechanisms that are involuntary and visceral. So now that we've got characteristics and functions out of the way, let's now get into the structure of skeletal muscle. We will come back to structure at the very end of this chapter, give a brief nod to the structure of cardiac muscle and the structure of smooth muscle so that we can apply some of the ideas that we're learning through the rest of this chapter to them. But from now on, we're mainly focusing on skeletal muscle structure and function. So skeletal muscles are bundles packed into larger bundles, packed into larger bundles. So bundles within bundles within bundles. Okay? So the entire muscle itself, one muscle, is considered the organ. So that one organ has a epimysium connective tissue surrounding it. That is what defines that muscle. Everything that is contained within that epimysium is that one muscle. So deep to that epimysium, you have more bundles called fascicles, and each fascicle has its own surrounding of connective tissue called perimysium. And then within each of those fascicles, you have little bundles, your muscle cells or muscle fibers. So individual muscle cells are packed into bundles called fascicles. Each muscle cell has its own surrounding called endomysium. And then within those muscle cells, you have even more bundles called myofibrils, Fibril. And those myofibrils are what contain the rope-like protein fibers that are going to create our actual contractile units, that when they pull on each other and shorten themselves, that is going to shorten the entire muscle. And those contractile units are called sarcomeres, what you see down here, which are composed of overlapping thin and thick filaments. This idea of overlapping filaments that pull on each other to shorten that length, that is a contraction. And if you have stimulated contraction through all of the sarcomeres in that muscle, then you can shorten it to its shortest length, give it its maximum tension. So bundles within bundles within bundles. If we work backwards now, again, the contractile unit is that sarcomere. Multiple repeating sarcomeres are organized into a myofibril, like a thin string. There are multiple myofibrils within one muscle cell, or aka muscle fiber or myofiber. Each muscle cell is surrounded by a connective tissue layer called an endomysium. Multiple muscle cells with their own endomysium are packaged up into these bundles called fascicles. Each fascicle has its own layer of connective tissue surrounding it called perimysium. And then multiple fascicles are bundled up together into one full muscle, one organ. And that muscle is defined by the epimysium that surrounds it, another layer of connective tissue. So a reminder about these prefixes that I've pointed out before in this class: epi, meaning around or outside, peri, meaning between, and then endo, meaning within. And then you have the root referring to what type of tissue we're discussing. So meso or myo are common prefixes related to muscle. We're also going to see sarco as a prefix related to muscle. So if you see any words with that myo, mys, or sarco as a part of their name, they're referring to muscle. Right. Here's another look at that same structure. Now, if we were to take a transverse section, right, cut all the way through that muscle, we can see these bundles in kind of a different way. So again, epimysium surrounding the entire organ, the entire muscle. You see all these bundles, these fascicles with blood vessels penetrating into some of the space between. Each fascicle has its own perimysium. Within those fascicles, we have individual muscle cells with their own endomysium separating each other. And then within each muscle cell, these tiny little circles are each a myofibril, which are what contain those sarcomeres, the actual contractile part of these cells. So we end up with these organizations of extremely long muscle cells. Muscle cells are some of the largest cells of the body, along with neurons. They are very, very long. So end to end, for any muscle that you're thinking about, all the way from where it attaches to the tendon on one side to the tendon on the other side, one muscle cell runs that entire length. Okay? So they go all the way. They are not, they don't stop and connect to another muscle cell and stop and connect to another muscle cell. Each muscle cell goes the entire length of the muscle and just has a bunch of these repeating sarcomeres that help shorten and contract each part of the muscle. So these tend to contract all together. So again, you might hear multiple names referring to a muscle cell. So muscle cell is synonymous with myofiber or muscle fiber. Any of those three terms show up in the textbook referring to the same thing, a muscle cell, or you might see them show up in different contexts, YouTube videos, any other articles you might be reading. But those three terms are synonymous. They all are referring to one muscle cell. And we have some unique terms for the structures of that muscle cell. So similar to what you were seeing with bone, right? Osteo as a common prefix referring to bone tissue. Right? Like I was pointing out, myo and sarco, in this case, referring to parts of a muscle cell. So Each muscle cell's membrane is referred to as the sarcolemma. We're going to see lemma, that suffix, show up again when we get into nervous tissue with the neurolemma. So the sarcolemma is the membrane of a muscle cell. These membranes, the sarcolemmas, don't just stay on the outside of the muscle cell like we're used to seeing with most of the cells we've been talking about and studying. The sarcolemma actually is also going to penetrate into the cell. It like pushes down, creating these little T tubules, which we'll see a better diagram of in a second, these transverse tubules. That is going to allow for action potentials from a neuron to stimulate an action potential on the muscle cell, on the sarcolemma, and that action potential will travel all the way down into these T tubules, which is going to cause a reaction deep within the cell at these terminal cisterns, which we'll talk more about in a second. So that one T tubule, that one penetration down into the cell with the two terminal cisterns that it is surrounded by, that structure is referred to as a triad. So we have three things, two terminal cisterns, one T tubule, that forms our triad. Those triads are deep within the sarcoplasm. So the sarcoplasm is referring to the cytoplasm of the muscle cell. So the sarcolemma is the membrane, sarcoplasm is the space within the cell. Within that sarcoplasm, you've got multiple nuclei and many mitochondria, depending on how you train your muscles, and whether we're talking about fast twitch or slow twitch or intermediate twitch fibers, that is going to change the amount of mitochondria, but there are lots of mitochondria in your muscle cells, very important for generating the energy that these cells need for contraction. Within that sarcoplasm, you're also going to find myoglobin, which is the muscle cell's unique protein for storing oxygen, again, for help with that ATP production, since aerobic respiration produces much more ATP than anaerobic. Muscles having their own structure, allowing them to store oxygen rather than simply relying on oxygen to be brought to them through the blood, allows for help with that ATP production. So very similar to the globin protein that your red blood cells have, right? So, muscle cells have their own myoglobin. And then what's taking up the most space in your muscle cells are the myofibrils and the sarcoplasmic reticuli. So the myofibrils, like we were just talking about, are what contain those contractile units of protein, those sarcomeres, or those contractile units for the muscle, those sarcomeres. The sarcoplasmic reticulum is the endoplasmic reticulum of muscle cells. The sarcoplasmic reticulum is specifically structured to store calcium ions. And calcium is going to be really important for controlling muscle contractions. Right, so those overlapping fibers that I keep alluding to, these sarcomeres, they are only able to function, to do their contraction, their whole job, when calcium is available, when they are exposed to calcium. That is what starts this whole contractile process. So the sarcoplasmic reticulum is full of vesicles storing calcium. They will release that calcium when an action potential travels down the T-tubule and reaches the terminal cisterns, which are these edges of the sarcoplasmic reticulum that are carrying those vesicles. And so a quick nod here to how muscle cells are created to give you a better idea of how we end up with these really long, large cells that can generate so much force and power. Right? They have this big job to do. Muscle cells are formed by the combination, the fusion of multiple other cells. And so that is why we end up with a cell that has multiple nuclei, because it started out as individual cells with their own individual nuclei, but rather than staying separate, they are fusing their membranes and their cytoplasm and all of their internal components together. And so forming this much larger fused cell with multiple nuclei, lots of mitochondria, lots of cytoplasm, sarcoplasm, and a large membrane. And we can continue to add to these cells. We can make them larger by fusing even more of these satellite cells, these myosatellite cells. So you can't fully regenerate an entire muscle cell. If you were to just lose that entire cell end to end, there isn't a way for your body to completely remake that naturally. But we can build a existing muscle cell larger if it just has some tears and some rips in it by adding more of these myosatellite cells, right, to help lengthen it, provide more cytoplasm, which makes more space for myofibrils to form, which makes more contractile units and thus a larger, stronger muscle. So you can see that here. Right? So when looking at a microscope image, this one's super zoomed in, or one of these less zoomed in images, right? All those purple dots are those nuclei. So if you see multiple nuclei, that's a good sign you're looking at a skeletal muscle. And then these striations, those lines that you see, this organization here, is due to those sarcomeres, those actual contractile units, right? Which we'll talk about here in a second. Okay, so now if we cut into like a little chunk of a muscle cell, we can get a good look of those internal structures that we've been referring to, right? So we have essentially removed the endomysium from what we're looking at. So all we see surrounding here is that sarcolemma, the actual membrane of the muscle cell. And everywhere that you see these little holes, right, and we can see that structure here on the side as well, that is showing that penetration of the sarcolemma, that where it's pushing into the cell, creating these transverse tubules. So what looks like these little yellow tubes running through here, that is an extension of that sarcolemma, the transverse tubules. And so we have this large network of transverse tubules essentially going around all of the myofibrils, those little bundles of protein filaments. And each T-tubule is going to end up surrounded by the ends of a sarcoplasmic reticulum, which we also see wrapped around each myofibril. And the ends of those sarcoplasmic reticuli are those terminal cisterns where you have vesicles full of calcium. So the idea that we're about to jump into when we start looking at actual contraction is that an action potential travels down the sarcolemma down the membrane of these T-tubules until it reaches a terminal cistern where it will stimulate the release of that calcium. The calcium will flood into the myofibrils where it will interact with these thin filaments, which is going to allow for those two filament types in the sarcomere to connect to each other, pull on each other, and create a contraction. You can also see a bunch of all the mitochondria that are all throughout that muscle cell. So yeah, those are the major structures of an actual muscle cell here. And so now zooming into our smallest part, right, our smallest bundle, these myofibrils, and taking a look at the contractile units, these sarcomeres. So sarcomeres, again, are these overlapping filaments. We have thick filaments and thin filaments. The thick filament, shown in purple here, is not going to change size or shape. It has these little knobs, these myosin heads, that are going to essentially pull towards the center, this M line that we have here. That is going to act like the centerpiece, where we are pulling the thin filaments towards. And so the thick filaments will pull these thin filaments closer to the center, and that creates a contraction. And so thin filaments have a lot of different proteins in their structure. So our main protein of focus, I guess to start with, is these little circular G-actin or globular actin proteins. You could think of these as like a magnet, where this yellow portion of the globulin actin is the active site, and it is trying to, or it wants to attract to something on our thick filaments. But in its resting state, it is not available. The active site is blocked by a different type of protein called tropomyosin. So this green intertwining rope-looking protein is covering those active sites, essentially making that magnet unavailable, so it cannot attach to the thick filament and cannot be used for contraction. And then we have troponin, which are these brown-looking proteins here, which are kind of acting as the control mechanism for tropomyosin. troponin determines whether tropomyosin is blocking those active sites or it has been moved away from the active sites and allows for the thick filament to interact and contract. So calcium is going to bond with troponin. That causes troponin to change shape, which is going to move tropomyosin out of the way, and that is going to expose those active sites on the G-actin so that they can be used for contraction. So those are the main proteins of focus. We also have this nebulin protein, which is creating kind of the structure. It is this strand in the center, which the globulin actins are surrounded by. And then we have titin, which is this kind of like coiled spring-like protein, which attaches between thick and thin filaments on the ends. Essentially acts as kind of like a spring for them. Great. And now taking a look at the thick filaments, which, as the name suggests, they are thicker. The thick filaments are mainly made up of myosin protein with these myosin heads, which are what are going to be generating force here for our muscles. These are what create all the tension in our contraction. So these myosin heads are on like this hinge, and they start in a cocked position. Once that troponin binds with calcium and moves tropomyosin and now exposes the active site of G-actin, that is going to allow that myosin head to attach to it. Again, like a magnet, they attract each other. And then that myosin head will power stroke, will push, right, along that hinge, like you see shown here. It'll bring that thin filament closer to the center, towards that M line. So on either side of this M line, we have heads in opposite directions. So if my wrists were the hinge and my hands are the heads, right, and my face here, my body is the M line, right? On the right side, we have heads that are going to pull towards the face. On the left side, we have heads that are going to pull in the opposite direction, but they both pull towards that center, towards the M line, and thus they are shortening the muscle, creating that contraction. So this is our resting state where we just have some overlap between these, but there's still space for them to move in the center, this H zone. And then when they are interacting, when calcium has been released and the myosin head can interact and pull those thin filaments, that is going to bring the thin filaments closer to the center, making that H zone smaller, okay? And that is contraction. And so essentially, the more overlap that we have between the thick and thin filaments, the stronger that contraction can be, right? If there's less overlap, there's less pull, and thus that muscle isn't shortening as much. And we'll talk about those things like tension and contraction strength later on. So that's all for this portion of the chapter. I will see you guys in the next one here shortly.